Okay, finally, protein molecules. General characteristics of proteins, they're made up of many amino acids, could be hundreds, could be thousands, depending on the protein. The protein molecule is a polymer. It is made of CH, O, and N, because if you look at the structure of an amino acid, it contains all of those. And then the R group sometimes contains sulfur as well. The backbone contains all of these things, but the R group may contain sulfur. And all proteins require a specific three-dimensional shape to function correctly. And that's because nearly all proteins need to bind to other molecules to function properly. And if it doesn't have the right shape, then it can't bind to that other molecule. So this is a generalized amino acid structure. You should be familiar with the structure of this amino acid. The amino group, NH2, the carboxyl or carboxylic acid group, the variable group, and the hydrogen. This is the only thing that changes from amino acid to amino acid. There's 20 different amino acids naturally occurring, and they each have a different variable group. Now, in nature, what usually happens is an amino acid that's floating around by itself will generally lose this hydrogen. It normally ionizes. It loses this hydrogen, and this picks up a hydrogen. So you may see that ionized form in diagrams. You may see NH3 plus and COO minus. That's generally how it exists in nature. To join amino acids together, you are going to join the carboxyl group of one to the amino group of another. So a polypeptide will be many amino acids joined together. One side will always be what we call the amino terminus, and the other side will be the carboxyl terminus, the C terminus, because it has this one-way orientation, right? So the polypeptide will be many amino acids in a row. Dipeptide would be two, polypeptide would be multiple, and then the protein is when it's finally functional and folded. For each new peptide bond you form, you have to remove a water. So if you're joining these 4 amino acids together, 1, 2, 3, you would have removed 3 waters. Okay, this is just another vision of that. So peptide bond is a covalent bond between 2 amino acids. So when you look at a protein chain, within the protein chain, these are all peptide bonds. And in order to break them down, you need to use hydrolysis. Enzymes are necessary for that, proteases. Some amino acids have hydrophobic groups, some have hydrophilic groups. The hydrophilic ones, sometimes they have no net charge, they're just polar. Sometimes they actually have a positive charge, sometimes they have a negative charge. But different amino acids have different properties. You can look this up in Google. In your textbook, there's a nice chart. So this is showing you that ionized form again, but you can see these would be basic because they're NH3. They've added, the backbone is always acidic and basic. It always has the negative and the positive. But the R group is what we look at when we're thinking about whether they're hydrophilic or hydrophobic, because in a protein, this actually will be joined together to another amino acid, and this one will as well. So those don't really play a role in the function of the protein itself. It's really the R groups that are doing that. So these are the basic ones, these are the acidic ones, and these are the polar ones. And these are all the nonpolar ones. So I don't expect you to memorize all of these amino acids. However, I would expect you to know by looking at an amino acid generally if it's hydrophobic or hydrophilic. So you'll notice all the hydrophobic ones are mostly C's and H's in their R groups because CH is a nonpolar bond. If you look at the hydrophilic variable groups, there's other things. Either they're charged, pluses, minuses, or they have OH. CO, those are polar bonds as well, and so CN and NH. So you're looking for those polar bonds, and that will tell you if it's hydrophilic or hydrophobic. This is just a picture showing you some unique 3D structures of different proteins, and the structure is going to determine the function. So if you change the structure, either by mutation or by denaturation, it's going to change I don't like to say change the function of the protein. It might disrupt the function of the protein. Right? Because when I say change the function of the protein, that implies to me that it's now going to do something completely new and different, which is possible, but it's more likely that it's just going to disrupt the natural function of the protein. All right, so protein folding. There are 4 levels of organization. There's primary structure, which is just which amino acids are there, right? It's serine, and methionine and valine and blah, blah, blah. Which amino acids are there in a row? Secondary structure is the partially folded polypeptide molecule. The tertiary structure is the fully folded polypeptide molecule. So both primary structures held together by peptide bonds, which are covalent. Secondary and tertiary are held together by hydrogen bonds mostly. But tertiary structure can sometimes be held together. There's also some disulfide bridges, sometimes some ionic bonds. These are covalent here. And so at this point, it's considered a protein molecule. Some proteins require 2 or more folded polypeptides to function, like hemoglobin has 4 subunits to function properly. So those are the different levels of organization. This is just showing you a diagram of that. So here is the tertiary structure. We're seeing there's an ionic bond, there's hydrophobic interactions, there's a disulfide bridge, there's hydrogen bonding. This holds that 3-dimensional structure in place. If that 3-dimensional structure gets disrupted in some way, that's denaturation, right? So hydrolysis is when the primary structure gets broken down. Denaturation is when the tertiary structure gets broken down. The folding, it's a loss of 3-dimensional shape, so that could affect the active site if it's an enzyme. This could occur because of pH, temperature extremes. Temperature extremes, basically the atoms are moving so quickly that they can't maintain those weak hydrogen bonds. pH, you're introducing hydrogen ions, OH ions, and those are going to potentially bond to, these other ones. So maybe this will now hydrogen bond with something else. And so that will disrupt the connections or interactions within the protein folding. Okay, and if you break down the tertiary structures in such a way that these covalent bonds, the disulfide bridges, break, when it renatures and goes back into its original form, the disulfide bridges may form differently, and it may actually be a problem, and it may misfold permanently. Sometimes protein denaturation is permanent, and sometimes it can be undone, depending on the protein. Not always. So an example of denaturation would be when you cook egg white in a frying pan, right? The proteins used to be present in that solution, and they were liquid and translucent. And when you cook it, they become solid and opaque, and we can't go backwards from that, right? So basically what happens is those proteins unfold, and then they cross-link with each other, and there's no going back from that in egg white anyway. I'm just going to go through this list of types of proteins. You can look at them more carefully in your PowerPoint slides. Just some examples: hemoglobin carrying oxygen around the body. Then you have cell structural proteins on the surface or muscle fibers. Enzyme proteins we're going to look more about later. They speed up reactions. Antibodies function in the immune system. Hormone proteins. Not all hormones are proteins, but some of them are, like insulin and glucagon. Growth factors, cell membrane, so cell transport and receptors. Okay, and then finally, enzyme function. I just want to talk briefly about this. We're going to get more into it a little bit later, but this reaction, this is showing a chemical reaction, and these are the substrates. These are the products, and it shows that uncatalyzed, that it takes a lot of energy. You have to put energy in to break these substrates apart, to break the reactants apart, and then they release energy when they form these new products. And this would occur without the enzyme, but not nearly as quickly. Sometimes it's like a billion-fold, right, difference. So if this is the enzyme, I want you to just notice here's the enzyme. The enzyme contains an active site, right? The active site is on the enzyme itself, and then the substrate comes in. The enzyme twists it and turns it into product, and it gets released. And notice that the enzyme is the same as when it started. So it can be recycled. It can be